Dear students, today I would like to talk about another lesson or another part of chromosomal basis of inheritance, the main event responsible for changes in the genetic material, that is a mutation. That's the next part. Okay, so we have genetic variations among the individuals that provide the raw materials which are the ultimate source of what we have, that is evolutionary changes. Normally mutation resulted in variations, and variations are the raw materials for evolution. So genetic variations among the individuals provide the raw material which are the ultimate what we have, the source of evolutionary changes. Now what are the causes of genetic variation? There are two main causes of genetic variation. Number one, genetic variation is due to mutation. This is number one. Another one, just a recombination process. The recombination process is a regular event happening during meiosis in all the sexually reproducing organisms. But the mutation is a rare one, occurs because of certain mutagenic agents in the environment or any alteration in the genetic material. So mutation, a rare event, whereas a recombination process is a regular event happening during meiosis in all the sexually reproducing organisms. And these two major or main events are responsible for genetic variation. What do you mean by mutation? A sudden change. All of a sudden a change occurs in the organism. A sudden change in the life of an organism. A sudden change in the genetic material of an organism is called what is known as mutation. A sudden change in the genetic material of an organism is called mutation. Now the concept of mutation was first studied by, that is, Hugo de Vries in 1901. While he has studied on, that is, we have a plant Inothera lamarckiana. When he was working with this, what is called Inothera lamarckiana, a plant commonly called evening primrose. It shows a number of alternative characters in it. So all of a sudden one character has been changed into another character. But normally, the mutation was first observed in the case of one breed of sheep. What we have, one breed of sheep by Seth Wright, I talked about while I was talking about that is in zoology. So Hugo de Vries normally coined the term mutation in 1901 and proposed what we have, the mutation theory, while working on a plant by name Inothera lamarckiana, commonly called evening primrose. Now we have changes in the genetic material. What are the main two broad types of changes? Two broad types of changes in genetic material. Number one, that is what we have point mutation or gene mutation. It's otherwise called gene mutation. Point mutation or otherwise called gene mutation. This is one of the changes in the genetic material. So what is point mutation? The mutational events that take place within an individual, the mutation events that take place within an individual gene is called what is known as point mutation. We'll discuss in later details. Then another broad change in the genetic material, chromosomal mutation, that refers to change in the number of chromosomes or change in the structure of chromosome. If there is a change in the number of chromosomes, we can say ploiding. Change in the structure of chromosomes, we can say chromosomal aberrations. We'll talk about later. So both together call as chromosomal mutation. Either change in the number of chromosomes or change in the structure of chromosomes, and that is called chromosomal mutation. Now, the compounds which induce, or the materials or the substances which induce mutations are called mutagens. We'll discuss in details also about this one, or mutagenic agents. Now these agents increase the rate of mutation. These agents increase the rate of mutation. And again you see that one, if you are taking these mutagenic agents, we have a number as I mentioned, we will talk about soon. Then the mutation may be a spontaneous one or induced one. This is based on the origin. The mutation may be a spontaneous or induced. The mutations may be spontaneous or induced. The meaning for spontaneous, naturally occurring mutations in the absence of mutagenic agents. Those mutations that occur in the absence of mutagenic agents are called spontaneous mutations, which occur naturally. And those mutations which are induced either by what we have physical agents or by chemical agents, they are called induced mutations. This is based on the origin. We can have actually spontaneous and induced. We have different types of mutations. We'll discuss one by one later. Okay, so this is about the spontaneous and induced mutations. So the process which induces mutations, the process which brings about the mutations, normally is called as mutagenesis. Mutagenesis. What is mutagenesis? The process or the phenomenon that normally brings about mutation is called mutagenesis. That is the formation of mutagens. And after that one, we have the resultant individual that is called as a mutant. After mutation, the individual is called mutant. So, the process of production of mutant varieties, okay, and that process is called mutagenesis. And actually, and the mutant organisms are collectively called as what's actually the mutagenic agents bring about normally mutations. The process of actually formation of mutant varieties is called mutagenesis. And the organisms after what is called after mutation are called simply mutant organisms. And those organisms are mutagenicized. And the organisms are said to be mutagenicid. So we have mutagenesis, the production of what's called the mutant varieties, and the organisms after mutation are called mutagenicid. We can say mutagenicid. And the organism after mutation is called mutant. We have mutagenicized, mutagenist organisms, and those are called the mutant varieties. So these are the different definitions. Mutant variety is different from the wild variety. The normal variety is called wild variety without any mutations. After mutation, the organism is called mutant. The agents which cause mutations are called mutagens. The organisms are said to be mutagenicid. So we are using what's mutagenesis instead of this S. I am using the letter S. Mutagenesis. Okay, so mutagenesis. This process is called mutagenesis, and this is what we have mutagenesis organisms after mutation. So this is about what we have. Now we have to go for the point mutation and also the classification of different types of mutations. Classification of different types of mutations. For example, based on the origin, either spontaneous or induced, based on the nature of the cells in which occurs, and based nature of the function, based on the nature of the molecular structure, based on the process of what we call translation. We have different methods that are adapted to classify what we call the mutations. Okay, so I'll take that one in the form of tabular column. I'll explain and then go for what is called actually the point mutation. Or I'll take for the point mutation and then just give the tabular column. Either way. Okay, now we have to go for what is point mutation. So we have to take the types of mutations, one point mutation, another one chromosomal mutations. First, I would like to take the point mutation. I mentioned already point mutation. Mutations affecting the single base of DNA or base pair of DNA. Mutations affecting a single base or base pair of DNA. Normally you know that one, just you see that one, point mutations. So normally the mutations altering the number of copies in a small repeated nucleotide sequences within a gene. Number of actually copies of small repeated sequences. That's a nucleotide sequences within a gene. That's what's happening in the case of gene mutations. That is a simple one, affecting what is called single base or base pair of DNA. They are called actually the point mutations. But I'll give a different, just a single definition as given in the book, and then go for the different types of what we have the point mutations. Now let's start with point mutation. Point mutation refers to the alteration of a single base pair of DNA or alteration of a small number of adjacent base pairs. That is called what is known as a point mutation. Okay, so the point mutation is broadly classified into two. One, base pair substitution, another one base pair insertion or deletion. Now what do you mean by base pair substitution? So it is a mutation where there is a change in DNA such that one base pair is replaced by another. That's a substitution. Replacement of one base pair by another base pair is called what is known as substitution. Okay, this is what is known as base pair substitution. What do you mean by base pair insertion or deletion? It is actually the addition or deletion of what is called the base pairs or nucleotide pairs. Just actually. Hence called base pair addition, base pair deletion. It is collectively called indel mutation by just adding the first two letters and three letters in the second one. Hence together called as indel mutation. So the base pair insertion or deletion otherwise called indel mutation. Now if you are taking the base pair substitution, it is of two types. What is called transition and transversion. What is transition? A base pair is replaced with a different base pair. One base pair is replaced by another base pair. Particularly, a purine base is replaced by another purine base. Or a pyrimidine base is replaced by another pyrimidine base. That's why I use the word different. A base pair in DNA is replaced with a different base pair. That is, purine is replaced by another purine. That is, A is replaced by G. These two are the purines. You know that one we already studied. Or a pyrimidine is replaced by another pyrimidine. Purine is replaced by another purine. That is, A is replaced by G. Or a pyrimidine is replaced by another pyrimidine. For example, T is replaced by C or C is replaced by T. Either way we can see. Either T is replaced by C or C is replaced by T. Here A is replaced by G or G is replaced by A. This is what's called transition. Same basis, what we have the purine basis. Exchange between purine bases or exchange between pyrimidine bases. Now the another type of base pair substitution is transversion. Here a purine base is replaced by another pyrimidine base, or a pyrimidine base is replaced by a purine base. Here purine to purine, pyrimidine to pyrimidine. Here purine to pyrimidine or pyrimidine to purine. So different actually the combinations. Now here transversion, a purine is replaced by actually a pyrimidine. For example, A is replaced by T. A is a purine and T is what we have pyrimidine. Similarly, G is purine replaced by C. That is what we have pyrimidine. Or that's what we have, A is replaced by C or G is replaced by T. This is the same combination, purine is replaced by pyrimidine. Or we can say a pyrimidine is replaced by purine. So pyrimidine T is replaced by A or just C, that is pyrimidine is replaced by G, or we can say another way, C is replaced by A or T is replaced by G. These two are what we have the pyrimidines and these two are the purines. Like that here these two are the pyrimidines and these two are the purines. That's about what we have the base pair substitution. I mentioned only the base pair insertion and deletion, either addition or deletion of nucleotide pairs. Hence called base pair deletion or base pair what we have just actually addition. And together called as indel mutation. Okay, this is first category. Okay, let's continue further. Actually what's happening, both the substitution and indel mutations affect the translation process. And based on this, as they affect the translation process, based on the translation process affected, the mutations are of the following types. And before that I would like to show first one, a normal DNA molecule without mutation. You know the DNA is a double helix, the two strands are antiparallel. One strand is running in 5'3' direction and another strand is running in 3'5' direction. Now they have the message in the form of genetic code, a triplet code. Now one of the strands of DNA is called as the coding strand having the message. Another strand is acting as a template strand. The message in the template strand is normally transcribed. Now 3'5' is the template strand. Now after transcription process, what's happening? The messenger RNA is synthesized in 5'3' direction. So the bases in the template strand are complementary to the bases of mRNA, except that in place of T we have U, because T is absent in mRNA. Now here I am taking the sequences TGT, TCA, CCA, T, and ACA, AGT, GGT, A. I represented the blood. What we have black color letters. There only we have to show the changes. Okay, either in the case of silent mutation or nonsense mutation or missense mutation or frameshift mutation. What's happening there? Now this is a messenger RNA having the base sequence EGU, UCA, CCU. So a triple codon is undergoing the translation process to form what we call the amino acids in a protein molecule. Now here EGU represents threonine, UCA represents serine, and then CCU glycine. So this is the normal DNA molecule, then transcription process, translation process. Now we have to go for what are the changes taking place with reference to the middle letters, what we have TCA, AGT, and here accordingly we have the following types of mutations. Number 1, synonymous or silent mutation. Synonymous or silent mutation. Here the mutation changes a codon for an amino acid into another codon for the same amino acid. A mutation causes a change in a codon. That codon is for an amino acid. That new codon also codes for the same amino acid. Now here and that, actually that type of mutation is called synonymous or silent mutation, because we didn't observe whether mutation occurred or not, because there is no change in the structure of the proteins. Now we started with the threonine, serine and glycine here. Then even after mutation we see that one TCG, and this is what we have TCG. It has been changed into AGC. So one of the bases what we have C has been changed. One of the bases what we have here TCG, and here also TCG, that's what we have AGC, and that is changing here. Instead of TCA we have TCG and AGC. This is a main letter which has been changed. You see that one. Instead of what we have AT pairing, we have GC pairing. But there is no change in the formation of amino acid. This sequence, like that of what we call the original, what we call the DNA molecule, is also responsible for the synthesis of the same amino acid serine. That's why we can say there is no external expression, a change in the phenotype, even the mutation occurred, hence called silent mutation. Mutation took place, but there is no change in the phenotype, namely the structure of the protein. And such a type of mutation is called synonymous or silent mutation. Okay, this is number 1. Let's go for the second one, non-synonymous or missense mutation. Here, a mutation which changes a codon for an amino acid into another codon for another amino acid. So one codon has been changed into another codon. That codon is normally for another amino acid. So a mutation in which a codon for an amino acid changed into another codon for another amino acid. Now I am taking here TTA. This is what we have actually, TTA. That is what we have in this case, TTA, TCA, like that we have. Now what happens after that one? This is the original one. Here just I took only after mutation. Now in place of what is called C, we have what is called T. And in place of what we have G, we have A. So the entire codon has been changed. Instead of TCA, we have TTA. Instead of AGT, we have what is called AAT. As a result, there is a change in amino acid. So one codon, that is TCA, has been replaced by another codon. As a result, the amino acid has also been changed in that protein molecule, and this is called non-synonymous or missense mutation. Simply, a codon has been replaced by another codon with a different amino acid. Here the original codon TCA, but here TTA. And here AGT, we have AET. As a result, this codon is for what is called leucine. So this is one, this is another one, This is the third one. So only I represent the red one just normally. The codon which is being changed resulted in leucine in that protein molecule. So these are the two types. We have to go for some more types related to what we have, the translation process, the mutations which affect the translation process. Accordingly, we are taking that one. Okay, the next one what we have to go for, what we have simply termination mutation or what we have nonsense mutation. What's happening there? Let's see the picture. Now the next type, nonsense mutation. So the mutation in which the codon for an amino acid is changed into a stop codon or termination codon. Then it's called nonsense mutation. A mutation without any meaning, because the stop codons are concerned with the termination process of what we have transcription. So a mutation in which a codon for an amino acid is changed into a stop codon, then it is called nonsense mutation. Now the next type we have the frameshift mutation. What is frameshift mutation? Mutation in which addition or deletion of a single base pair occurs. Addition or deletion of single base pair occurs. As a result, what is happening? The reading frame of the translation process has been changed. The reading frame we already studied in zoology, the sequence. The reading frame for translation process has been changed. As a result, there is a complete loss of what we call the protein structure and function. Then it's called frameshift mutation. It may be an addition. Here just you see that one, a base C is added in place of what we have E. A base C is added in place of E. So the shifting occurs because of the addition of C. What happens? The shifting occurs towards the right side. As a result, two bases or nucleotides are left because of the addition of one. Okay, that is one, that is addition. Now in the case of deletion, here the base T has been removed. So we have, you know, that one TCE, the T has been removed. So the shifting occurs from, that is right to left, or shifting occurs towards the left side, so that we have the combination has been changed CAC and GTG. So in this case, the first one there is no change. Because of the shifting of the base towards the right side, because of the addition of a base, that is C, what will happen? We have glutamic acid instead of what is called the serine, and also arginine in place of what is called glycine. Now in this case, we have, that is threonine, no change. Because of the shifting of the base towards the right side, towards the left side, what happened? This is because of the deletion of the base, either T, I put minus, the meaning for that one, deletion of T, minus A, deletion of A. So the shifting occurs towards the left side. As a result, we have the bases have been changed, the amino acids have been changed. Now here we have valine and valine instead of what we have just serine and glycine. We are getting valine and valine. This is because of the change in the reading frame. That's about the deletion mutation, addition mutation. In addition, shifting occurs towards the right side. In deletion, the shifting occurs towards the left side because of the removal of a particular nucleotide, and here the addition of nucleotide. Here the added nucleotide is C, that is C, here the G. Here the removal nucleotide is T, and then A here, the next strand. So in such a way, we have what happens now, the mutations occur. Now we have to go for two more. Under the substitution mutation, there are two types of substitution mutations I mentioned earlier, transition and transversion. I will complete that one with the diagram. So far we have studied that is about the mutations which affect the translation process. Now I already mentioned about what I mean by substitution mutation. Here one base is substituted by another base, or one base pair is substituted by another base pair. There are two types of substitution mutation I already mentioned about. One is transition mutation. In transition mutation, one purine is replaced by another purine. Purine is replaced by another purine, or one pyrimidine is replaced by another pyrimidine. So the purines are, you know that one A and G, the pyrimidines are T and C. Here we see that one, that is A, is found in the normal DNA molecule. It has been changed by G, that is nothing but another purine. So in a normal DNA molecule, in that position we have adenine, that is a purine, that is being replaced by another purine, that is G, what I represented here. So purine is replaced by another purine, or a pyrimidine is replaced by another pyrimidine. Now you see that one T and C are the pyrimidines. Now here what is happening, the T has been replaced by C. That is called transition. Either purine is replaced by another purine or pyrimidine is replaced by another pyrimidine, then it's called transition. Now what is transversion? In transversion, a purine is replaced by another pyrimidine, or a pyrimidine is replaced by a purine. So adenine and guanine are the purines, cytosine and thymine are what we have the pyrimidines. Here you see that one, in the place A, now this is a normal base present in a normal DNA. This is what's called a purine, which is being replaced by a pyrimidine, what's called the thymine T, as we represent in red color. So normal position we have adenine, a purine, which is replaced by a pyrimidine, what is called thymine. Likewise here you see that one, in a normal place we have thymine, just what we have the pyrimidine, which is being replaced by a purine, what is called the adenine. So here either purine is replaced by pyrimidine or the pyrimidine is replaced by purine, and that process called transversion. That means A is replaced by T or C, G is replaced by T or C. Then it's called what we have transverse mutation. So these are the different types of mutations. This is based on what we have either translation process affecting mutations or based on substitution. Now we have to go for the different types of mutations based on the cells in which occurs, whether induced or that is anything else. Based on the molecular structure we have completed. Then we have to go for based on the functional aspects. I'll discuss that one in the next class tomorrow. Okay, thank you. Goodbye.